All right, welcome to This Explainer. Today, we're diving straight into the invisible war against pathogens, breaking down the exact science and the crucial stats behind the machines that keep our hospitals safe. Whether you're prepping for medical exams like the NEET PG, or you're just genuinely fascinated by how modern medicine actually manages to keep microscopic invaders at bay, you are definitely in the right place. Here's our blueprint for today. We're going to start with the absolute baseline of cleanliness, look at the power of heat, break down the hot air oven versus the autoclave, put them head to head, and then wrap things up with chemical disinfection. Let's get right into it. Section 1, the absolute state, sterility. So, can an object be partially sterile? I want you to really think about that for a second, because in a clinical or hospital setting, the answer to that is literally a matter of life and death. We throw around words like clean, sanitized, or disinfected all the time, right? But in the realm of microbiology, words have very strict, unforgiving meanings. Take sterilization, for example. It is the complete destruction or removal of all forms of microbial life. We aren't just talking about your standard everyday vegetative bacteria here. We're talking about fungi, the absolute hardest to kill viruses, and most importantly, highly resistant bacterial spores. This process leaves an article completely 100% free from any viable microorganisms. It's the absolute sealing of clean. The big takeaway here is that sterility is an absolute state. To put it bluntly, and you'll hear this joke in hospitals, claiming a surgical instrument is partially sterile is exactly like saying someone is partially pregnant. It just doesn't exist. It either is or it isn't. If even a single resistant spore remains, that instrument is not sterile. Period. Understanding that absolute sealing really helps us define everyday terms. While sterilization kills absolutely everything, disinfection and antisepsis have totally different jobs. Disinfection? That's strictly for inanimate objects. Think hospital floors, walls, furniture. It destroys most pathogens, but yeah, it might leave those stubborn spores behind. Antisepsis, on the other hand, involves chemical agents like povidone iodine or alcohol rubs. That's what we safely apply to living tissue, like your skin before surgery, to inhibit microorganisms. I mean, you wouldn't sterilize your skin because that would severely damage the tissue, but you absolutely want your surgical tools sterilized. Moving on to Section 2, the power of heat. Now, let's think about the contrasting mechanisms between the two main types of heat we use. Moist heat is super efficient. It works by rapidly coagulating and denaturing proteins. It's exactly like boiling an egg. The heat transfers instantly through the water, permanently altering the proteins inside. Dry heat, though, is a much slower process. It destroys microbes mostly through slow oxidative damage and dehydration. You are literally slowly baking the germs until their cellular constituents oxidize and break down. Since it lacks moisture to transfer that heat, dry heat requires way higher temperatures and much longer exposure times to do the job. Which brings us to Section 3, baking germs, the hot air oven. If you are taking any medical exams, memorize these critical parameters. For the hot air oven to actually guarantee true sterility, it requires this exact combo: 160 degrees Celsius for 2 full hours. And here's the kicker. That 2-hour timer, it only starts after the entire chamber has reached 160 degrees. So yeah, it's a slow burn, but it's absolutely crucial for the right materials. So what are those right materials? Well, the oven is perfectly suited for moisture-sensitive items. Because it uses dry heat, it's brilliant for glassware, powders, oils, and sharp metallic instruments that might otherwise rust in steam. But there are strict restrictions. You absolutely cannot put rubber, heat-sensitive plastics, or liquid culture media in there, or they'll be completely ruined. Oh, and definitely remember its specific biological indicator, Bacillus atrophous. We run these highly resistant spores through the oven. If they die, we know for sure the machine is truly sterilizing. All right, Section 4, under pressure, the autoclave. Now, we're talking about the undisputed champion. These are the golden numbers for the autoclave. 121 degrees Celsius at 15 pounds per square inch of pressure for 15 to 20 minutes. Now, wait a minute. Water normally boils at 100 degrees Celsius, right? Well, that's exactly where the pressure comes in. The pressure itself doesn't actually kill the bugs. It's just there to allow the water vapor to get superheated, up to 121 degrees. That specific combo of heat, pressure, and time is the magic formula needed to obliterate even the toughest bacterial spores through rapid protein coagulation. The absolutely most crucial part of this whole process, removing all the trapped air.
See, when the autoclave heats water to generate steam, that steam has to displace the air. If you don't actively vent and remove that trapped air, you get an air-steam mixture, and air blocks the proper penetration of steam. That means the lower portions of the chamber won't reach that magical 121 degrees, and your instruments stay unsterilized. The air literally has to go.
Like I said, the autoclave is the gold standard for microbiology labs and hospitals. It's perfect for culture media, surgical dressings, rubber goods, most surgical instruments, you name it. But it does have its kryptonite. It cannot handle oils, powders, or fat-containing substances, simply because steam can't penetrate them. And to prove the autoclave is doing its job, we use a different biological indicator, Geobacillus stereothermophilus. Unlike the last indicator we talked about, these spores actually love the heat. So if the autoclave can successfully kill them, it can literally kill anything.
Section 5. The ultimate showdown. Okay, think of this as your ultimate high-yield cheat sheet for exams. There's a beautiful symmetry here. The autoclave uses moist heat for rapid protein coagulation in just 15 minutes. The hot air oven uses dry heat for slow oxidative damage over 2 solid hours. The autoclave uses Geobacillus stereothermophilus. The hot air oven uses Bacillus atrophilis. Understanding these differences tells you exactly which tool to reach for in a clinical setting based on what material you need to sterilize.
Finally, Section 6, Chemical Warfare, Disinfection. Now, as we look at chemical agents, keep this big limitation in mind. While disinfection is fantastic for general safety and controlling disease spread, it is fundamentally a step below sterilization. Why? Because those stubborn bacterial spores can and will survive it. That's why we only use this for environmental surfaces or instruments that don't penetrate sterile body tissues.
Let's quickly break down the common chemical weapons we use. Alcohol is our go-to for standard skin disinfection, something we're all pretty familiar with now. Chlorine is for water. Phenols are fantastic for cleaning hospital floors and surfaces. Then you've got the heavy hitters, like glutaraldehyde. That is absolutely crucial for high-level disinfection of delicate equipment, like endoscopes, that would melt in an autoclave. And lastly, formaldehyde is what we use for fumigating entire rooms. Before we wrap this explainer, let's rapidly hit these critical, high-yield takeaways. Remember, moist heat will always be more efficient than dry heat. Match your material to your machine, oils and powders go in the oven, culture media goes in the autoclave. Tyndallization? That's a fractional method taking 3 successive days of steaming. And here's a fun fact that surprises a lot of people. Despite what many consumers think, pasteurization does not actually achieve true sterilization of your milk. It kills the harmful pathogens so it's safe to drink, sure, but it leaves some harmless bacteria behind, which is exactly why your milk eventually spoils if you leave it in the fridge too long.
I'll leave you with this provocative thought as we wrap up. If even a single, highly resistant bacterial spore survives on a surgical scalpel, is the patient truly safe? The answer, of course, is no. And that right there is exactly why understanding the absolute, uncompromising nature of sterility and mastering these machines is the very foundation of modern medicine. Thank you for joining me on this deep dive into the invisible war. Keep asking questions, keep studying those parameters, and I'll catch you in the next explainer.